Hi, everyone. So I'll talk a little bit about enzyme kinetics. Oops. Yeah, sorry. Problem is. Okay. All right. So enzyme kinetics, and I want to make this presentation so you can see it, and this will go along with your enzyme kinetics homework. So we need to finish this off. This is what we didn't talk about in class today. So I just want to tack this on. So enzyme kinetics of a reaction are simply just the rate of the reaction and how it changes in response to different environmental stimuli, right? So it's a useful thing to do to know or understand, because you can actually measure the effect of a small molecule on an enzyme by measuring its kinetics, right? So the rate, if you want to inhibit an enzyme, then if you decrease the rate of that function of the enzyme with a small molecule, then you know that small molecule function is an inhibitor, right? And so that maybe it functions as a good drug against that enzyme, right? So one of the earliest ways of doing this with enzyme in enzyme biology was the development of Michaelis-Menten kinetics, the names of the two of the scientists who worked on this. And they came up with an equation to relate substrate concentration, increasing substrate concentration with velocity. So velocity is basically a rate, right? So it's amount of substrate used or product produced per unit time, right? That's V. And substrate concentration, of course, is just S, increasing amounts of substrate. So if you take run a reaction with constant enzyme, right, but increasing substrate concentration, right, and you use constant time and run the reaction, right, you'll produce product or use substrate at a certain rate, at a velocity, which is amount of product produced or substrate used per unit time, right? Okay, now any particular reaction, the increased substrate concentration, eventually the reaction will reach a maximum velocity. At some point, you'll increase the substrate concentration to the point where you can't actually increase the velocity anymore. And you should, in the next few minutes, think about why that might be. We'll talk about that in a second, but think about why that might be. At some point, you're going to reach a substrate concentration where the velocity cannot be increased any further, right? And that is what's called the Vmax. It's a maximum velocity of reaction. It's kind of a constant, not a strict constant, but it's a constant for any particular reaction that you can measure, that measures the effectiveness of an enzyme, the maximum velocity an enzyme has, right? Under any particular conditions, right? So that's one term we need to be aware of. The second term we need to be aware of is this thing called the Km, which is derived from taking the measurement of half Vmax, the velocity at half Vmax, and then finding the substrate concentration at that half Vmax. So that would be right here, and that's called the Km, the Michaelis-Menten constant, right? Michaelis-Menten equations is this one: V equals Vmax over substrate concentration, or multiplied by substrate concentration, divided by Km plus substrate concentration. This is a very hard equation to work with, and this is all, of course, done in the 40s and 50s before computers are common. So this is hard to manipulate. But it does relate velocity to Vmax and Km with increasing substrate concentration. So this is called Michaelis-Menten kinetics, and this is what the curve looks like if you do this kind of measurement in an enzymatic reaction. Okay, so Vmax represents the maximum velocity of the reaction. Of course, what is Vmax then? Vmax occurs when all the enzyme is occupied by substrate. In other words, each enzyme molecule is functioning at full speed. It can't go any faster because it can't take any more substrate any faster in any reaction cycle, right? So Vmax is maximum. Every enzyme is going at full speed, right? Km represents the substrate concentration S at which a reaction proceeds at velocity equals half Vmax. Keep in mind, Km is not half Vmax. It is the substrate concentration at half Vmax. Km tells you about the affinity the enzyme has for its substrate, right? A low Km represents an enzyme with a high affinity for its substrate. In other words, the enzyme binds to its substrate well, right? So it really interacts with substrate effectively. A high Km represents an enzyme that has a low affinity for its substrate, right? So it reacts less well. So that would look like, so a high Km enzyme would have a steeper increase in velocity and then flatten out to Vmax. So the Km would be closer here. And a low Km reaction means that the enzyme reacts with substrate very effectively. So it interacts with substrate well. a high KM reaction would be flatter, right? Run out like this, and the Vmax would be here, would be the KM would be out here somewhere, so the KM would be higher, right? And that means an enzyme is a little more sluggish interacting with substrates. It doesn't reach half Vmax as effectively at high substrate concentrations. All right, so this equation is a pain and hard to work with if you're trying to solve for Vmax or KM. So what we're going to do, and what Leidenweaver and Burke did, it's two scientists who work on this, is they took the reciprocal, of the Michaelis-Menten equation, literally 1 over V, right? 1 over V equals 1 over this thing, and they solved it for 1 over V. They solved this part and related to 1 over V, and they found that that is 1 over V equals KM over V max times 1 over substrate concentration plus 1 over V max, right? You want to actually get this so that you have velocity related to substrate concentration, some sort of way that allows you to isolate V max and KM. And so this allows you to do that. This should look familiar to you. This Y, right, equals something times a term, which is on your what's going to be on your x-axis, right? So that could be Y equals A times X plus a term, right? 1 over V max. This would be a slope. So if you do this, if you take your reactions, you run increasing substrate concentration with constant enzyme, and you run them, you actually literally run the reaction, and you measure the velocity of each of those different substrate concentrations, then take the reciprocal of the velocity and plot it against this reciprocal of the substrate concentration. The substrate concentration, 1 over substrate concentration on the x-axis, 1 over velocity on the y-axis, you will get a linear relationship, a straight line. The slope will be Km over V max here, and the y-intercept will be 1 over V max, right, B. Okay, so now when we have 1 over V, these terms, and 1 over S, these terms, we can actually figure out what Km V max is with these, using this equation, right? So this actually becomes pretty easy to work with. All right, so you can use this now to solve kinetics problems, solve what Km is and V max, right? And so you're going to do an enzyme kinetics experiment, sorry, not experiment, a problem set that's going to allow you to work, ask you to manipulate this and work with it, right? And solve Km V max different equations. Now, why do we care? Why is this important? All right, well, you can measure the effect of inhibitors on enzymes by using these kinds of equations, right? Doing these kinds of experiments. So inhibitor, we already talked about, this is a substance that blocks the ability of an enzyme to catalyze a chemical reaction. Now remember, there's two main types. There's competitive inhibitors that compete with the substrate to occupy the active site, but they're not stable usually, right? So most competitive inhibitors are interacting with weak interactions, and they can actually fall off the active site, and substrate can slip in there, right? So the more inhibitor you have relative to substrate, the more inhibited the enzyme is going to be, right? But if you add lots and lots of substrate, at some point, the competitive inhibitor is swamped out by substrate. You can't have access to the active site because there's so much substrate around that the concentration of the inhibitor becomes basically negligible, right? So large amounts of substrate will overcome inhibition of a competitive inhibitor, right? So this means that these inhibitors, competitive inhibitors, raise the Km of the reaction, but not the V max, right? Because as you increase the substrate concentration, the reaction will approach V max, even though the competitor is still there, because you've basically swamped it out and it's no longer able to inhibit enzyme anymore. So the V max stays constant. What changes is that the reaction slows down. So the rate of the initial rate of the reaction, or essentially the effect is the affinity of the enzyme for substrate is decreased because the competitive inhibitor is getting in the way, essentially, right? So the Km increases, right? For a non-competitive inhibitor, remember that's an inhibitor that binds somewhere on the enzyme other than the active site, right? And changes the shape of the enzyme so it no longer can function, right? So the active site no longer functions. So that means substrate concentration is not affect the inhibition of the, will not be affected by the inhibition of the reaction, right? So inhibitors will block, will change the structure of the enzyme and basically just remove that enzyme that's interacting with a non-competitive inhibitor from its ability to actually cause to catalyze reactions, right? So you're essentially decreasing the effective concentration of enzyme, which will mean that Vmax will decrease, right? So Vmax will decrease in these reactions, because you basically are effectively having less enzyme functioning, because non-competitive inhibitor is binding on the enzyme and inactivating it, and that binding is not affected by substrate concentration, right? So Vmax will decrease, but the KM will stay the same, and that's because the enzymes that are left behind that are not inhibited by inhibitor, assuming there's more enzyme than inhibitor around, right? They will still function like normal enzymes, so they'll have normal substrate interaction. So the affinity for substrate will be the same for the remaining functional enzyme reactions, and the KM will stay the same. But because it'll be less effective enzyme, the Vmax will decrease. All right, what does that look like? Here's a normal enzyme Michaelis-Menten curve in purple, right? The competitive inhibitor, it's flatter, right? But still the Vmax is reached at higher substrate concentration. So Vmax is reached out here somewhere with no inhibitor, here with inhibitor, right? Okay, so it's a little further, which means that the KM, Vmax is the same, right? But the KM, which is half Vmax, would be here for no inhibitor and out here for presence of inhibitor, right? Okay, now what about non-competitive inhibitor? So non-competitive inhibitor, Vmax will be decreased, but if you measure the KM for this reaction is going to be somewhere around here, right? Half Vmax, and Vmax is this new Vmax in the presence of inhibitor. So half Vmax will be around here. If we compare that to the normal enzyme in the absence of inhibitor, that half Vmax is somewhere around here. So it's about the same place. So KM has not changed. Okay, so this just shows us again another way. All right, so now why don't we plot this using Lineweaver-Burke plotting, right? So what happens, right? Well, here's a line of an enzyme with no inhibitor present, right? The slope is KM over Vmax, right? And the y-intercept here is 1 over Vmax, right? So I showed you before. Now, if the Vmax is staying the same in competitive inhibition, but the KM is increasing, the KM over Vmax, or the slope, is going to increase. So the line's going to increase, right, in slope, but the 1 over Vmax term's not changing. So the y-intercept stays the same. So with competitive inhibitor, you have a steeper line that goes through the same y-intercept, right? If you have a non-competitive inhibitor, now remember the Vmax is going to be decreasing, right? And remember the slope is KM over Vmax, so that's going to make the slope increase, right? And the 1 over Vmax is going to be increasing also because the Vmax is getting smaller, right? So the slope increases, KM over Vmax will increase because KM is staying constant, but the Vmax is getting smaller. So KM over Vmax will increase, and the slope is going to increase also. 1 over Vmax, if you make Vmax smaller, 1 over Vmax is going to get bigger, right? And so you get this kind of line where you have a new y-intercept and a steeper line. Non-competitive inhibition, all right? Okay, so they're easy to tell apart when you run the reaction. So now you guys are going to do enzyme kinetics experiments in a couple of weeks where you're taking alkaline phosphatase and running reactions and measuring the Vmax and KM of those reactions, right? And then you're going to take small molecules and see what the impact of those are. They function as non-competitive inhibitor, competitive inhibitor, not as an inhibitor at all, or are they even functioning as activators or doing something else altogether? So that's what you guys are going to try to be working on in the lab in the week after next, right? For now, I need you to use this information to help you solve enzyme kinetics problems that's been assigned to you over the weekend. And I hope this is going to have helped you do that. Don't worry about this. We'll talk about that later. And if you are interested, you can there's some additional problems. MIT Hypertextbook of Biology has some additional enzyme kinetics problems if you want to use them to do some practice. All right. I think I posted them on the Canvas page. Right. Okay, that's it. So good luck, and let me know if you have any questions.